Okay, thanks very much to the course organizers. It's a real honor to be here at the LA Live course at the 2022 SCSG GI Symposium. So I was asked to speak about single-use duodenoscopes. These are my disclosures. As you can see here, I'm a consultant to Boston Scientific, and I have done research on one of the single-use devices. So this is a summary of really how we got here. So in order to understand why single-use devices came about, we really need to understand the issue of duodenoscope transmitted infections. And they really began in the early 2010s, initially in Europe, and then in the United States. And there have been more than 45 sites and 350 patients infected. But this is really likely underreported because it's really hard to prove these cases. And we've actually helped many sites identify their outbreaks because of some special research our labs were doing at UCLA. And we've seen this with all manufacturer types in terms of these device-transmitted infections. In 2015 was definitely the peak in terms of reports, about 250 reports. And this has declined over time. And so we've made progress, despite being more aware, to under 100 reports per year in 2017 and 18. But there were still 3 deaths in U.S. patients in 2018 related to infections associated with these devices. You can see here the global scope of the problem from a paper we wrote about 4 years ago. You can see that it started really in the early 2000s, these device-related ERCP infections. You can see CRE in particular started out more in Europe, so you can see a lot of the early sites were in Europe, and then it sort of made its way to the United States in the early 2010s. And you can see both our hospital and Cedars had an outbreak around the same time in August and October of 2014. And again, this has continued over time, although to a lesser extent. And the FDA recently brought this back in the news again on April 5th because they requested that duodenoscopes with innovative designs to enhance safety should be, sort of utilized when available, and that they supported the transmission to these devices. And they did this in part because they had promising preliminary results from their post-market surveillance of disposable end-cap devices, which I'll discuss in a moment. So the advisory specifically said that use duodenoscopes with disposable components are fully disposable if you have them, and that they may lower but not necessarily eliminate the risk of infections when you have disposable components, of course. If you have a fixed end-cap duodenoscope, they encourage transitioning to these newer models, and they commented on the fact that some devices manufacturers are offering no-cost replacements, but they didn't provide a specific timeline for the transition. They also said follow your manufacturer's instructions for the assembly of the devices, and make sure you do routine inspection, and make sure your staff are well trained, and that you consider using supplemental measures as well to reprocess your scopes, which I'll get to in a minute. So it's not just, however, clinical infections that are relevant, it's also how often a device that we believe to be clean is actually contaminated. And so the FDA did a fixed end cap post-market surveillance study, and they were expecting a 0.4% contamination rate with pathogenic organisms, and they found it was 12 to 13 times as high, between 5 and around 5% for the 2 major models in the US. In addition, a human factor study is looking at how well people follow the steps of both in-room pre-cleaning as well as manual cleaning showed that 75 to 80% of the pre-cleaning steps were done incorrectly, and that 7 out of 8 elevator cleaning steps, at least 7 out of 8 times, there was at least one step that was done incorrectly, and in about a quarter of the cases, half the steps were done incorrectly in terms of elevator cleaning manually. So I think these devices are somewhat complex to clean. And this is not just a United States problem. The Dutch, which consolidate their ERCPs to 73 centers, invited all of them to provide 4 to 6 samples per their devices, and they all cultured centrally. 92% of the sites participated with 155 duodenoscopes and 745 sites sampled. And what they found was that 1 in 5 of their duodenoscopes were contaminated, and 15% actually had GI and oral organisms that were grown. And that about 40% of centers had at least one contaminated device. So again, a pretty substantial number. Now, as we know, this is the reprocessing cycle we currently use, high-level disinfection. Every time you see a blue arrow, it's a step. Every time you see a green arrow, it's for something to go wrong. and each of the things next to the green arrow are what it could be. And that could be due to device damage, it can be due to ineffective cleaning, the presence of soil, perhaps impaired reagent, inadequate drying, recontamination or biofilm. So lots of steps that can occur. So again, lots of opportunities. Now, why are duodenoscopes in particular so difficult? Well, it's primarily the elevator here. And as you can see that there's this recess behind the elevator. There's an elevator cable. There's a little V-notch for the wire mechanism. There can be a hinge mechanism here, divots on the scope. So there's lots of different things here that add complexity compared to regular scopes. And the reason that we run into these challenges is that the cleaning protocols to clean these devices have never been validated in clinical practice. It's usually lab testing. We sort of soil it with a certain amount of bacteria artificially and then sort of assess it in a limited number of cases. And the FDA has said that, as you've seen, that the user error is well known, but they've commented that user error is part of device design, and if something is too complex to clean, it's up to the manufacturer to make it more easy to clean rather than to blame the user. And again, HLD typically lacks a thin safety margin, especially for highly contaminated devices, and lacks sort of redundancy for levels of protection. And the other hard thing is there's no quality check. It's hard to have a visual cue to alert a user to error automatically, and there's really no final quality check on the entire process. People have tried using culture ATP, but they haven't correlated well with each other and aren't entirely reliable. Almost half the sites that had an outbreak had negative cultures on their scopes. So the FDA suggested, well, you can enhance your recleaning process. You could run the device through the AER, an automated reprocessor, a second time. That'd be double HLD. Ideally, you'd manually clean it twice as well. You could culture in quarantine. So you could culture after reprocessing, keep it aside until the cultures were negative, and then use. You could sterilize with either gas, ethylene oxide, or liquid chemical sterilization, usually peracetic acid. So, well, what should we do now? We recently wrote a multi-society task force article about endoscope reprocessing. And when we looked at sort of the high-level evidence for double HLD versus sort of single HLD, 2 randomized trials showed that there was no additional benefit compared to single HLD of double HLD in reducing bacterial contamination rates in duodenoscopes. Well, what about ethylene oxide? Well, in the non-outbreak setting, there was 1 randomized control trial, albeit granted limited data, that suggested ethylene oxide sterilization did not reduce bacterial contamination either compared to single HLD. Now, they had a very high contamination rate in all arms because they used single-person culturing, and so maybe methodologically there was an issue, but again, it did suggest that that maybe there's not a clear benefit. Now, we did comment that ethylene oxide did seem to have a role in helping terminate outbreaks when they occurred, and should be considered in those situations. And there was no role to suggest ethylene oxide sterilization for other types of scopes. So I've just told you 2 out of the 4 enhanced reprocessing techniques may not be enough. Well, what about disposable tips and perhaps disposable elevators? Well, that was the nature, the preliminary results were why you saw this April 5th advisory from the FDA. So I showed you that it was kind of around 4 to 5% with these, other devices, of the fixed end cap. And then they looked at the early data. They had half of the data from one of the manufacturers and found a contamination rate for high concern organisms only 0.5%. So this was about a 90% reduction compared to fixed end cap. Now, it was only half the data from one of the smaller manufacturers. We're still waiting for the 2 larger manufacturers to provide data, but I think they were excited to see this data and wanted to share it, which is why it came out. Excuse me. But it's not just the elevator. So there, again, there's a study out of the Pacific Northwest looking at 4 centers in the Providence system using 2,925 encounters, 45 duodenoscopes and linear echoendoscopes, and they daily cultured them for 6 months for both the elevator and the working channels. And they found a similar 5.2% contamination rate on the elevator, but they also found a 2.9% contamination rate in the channel. So it may be more than just the elevator, although it's probably at least the majority from the elevator. So that then suggests that if you can't just get it by using the tip, maybe perhaps we need to replace the whole device, and that was where the concept of a single-use ERCP scope or duodenoscope came about. And the pros of this are exactly as I mentioned. There's no issue with reprocessing inadequacy. You could define exactly how much your inventory was. So, you know, if you start today with 8, you'd have 8. You wouldn't have to worry about reprocessing. So they're typically lighter, so there may be ergonomic advantages, and perhaps they're customizable in the future. So right and left-handed versions or knobs of different sizes for ergonomics. The cons, of course, are, can we really afford this? These devices are usually well over $1,000, sometimes many thousands of dollars. How good are they in quality and performance compared to a reusable device? And will they be durable during, even if they're only for one case? What if it's a 2-hour case? Will they last? Are they strong enough and sturdy enough? And of course, what about all the medical waste that's generated? There are currently 2 devices on the market right now. On the left is the AMBU device, the single-use A-scope, and then the single-use Exalt Duodenoscope by Boston Scientific on the right. And there's been more data from the one on the right, so I'm going to share that data, at least in terms of published data, although there was some data this year's DDW from the AMBU single device. Again, these devices in general are meant to sort of mimic what currently is available. They're not expected to be dramatically different. You can see slightly bigger tip on the Exalt device, slight increase in field of view. It's lighter and a little bit, the staff a bit stiffer as well. So I had the opportunity to lead the first human use study of this device in patients. And we had spent quite a bit of time testing this in animals, so we did our first 13 cases just pushing the scope to the right spot, but we didn't actually do the ERCP, and then we did 60 for real. What's interesting is that we know that ERCP is associated with high rates of technical failure and adverse events, and we were using a device at the time was not FDA approved, but despite all of that, we found that out of the 81 patients that we approached, only 4 were not willing to or able to give consent. People were very enthused about a single-use device and gave permission. And the 13 roll-in cases all went fine. We were able to maneuver the GI tract, and then we did the 60 cases in which 58 were done successfully without crossover. There were 2 crossovers. We had 3 and 7 day follow-up, which was all comparable in terms of adverse events. So here's the data on sort of the types of patients we did. You can see that the majority of patients did have a prior sphincterotomy, about 2/3. Most of these cases were for stent placement or removal or exchange or evaluation of a biliary stricture, less often for pancreatic work or beduct stones, and in most cases were ASG grades 2 and 3. So the overall satisfaction of endoscopists with 10 being what they would normally get with their reusable device, it was about a 9, and we looked at all the different aspects, cannulation, sphincterotomy, stone removal, all the different maneuvers, and we actually came up with 14 specific maneuvers. We had 177 ratings, and 160 of this 177 were neutral or better. Only 2% of the time the single-use device was preferred, about 8% it wasn't. And when it wasn't, it was usually related to cannulation or stent placement or removal. This is a sort of look at the common things we do: biliary sphincterotomy, cannulation, clearance of stones, placement or removal of biliary stents, violation of strictures. And you can see all 90 to 100% approval ratings here. And again, this is another way of looking at it in terms of the yellow showing kind of how it feels out of the box. The white is sort of the maneuverability type issues. The green is how well does it cannulate, and the light green and the dark green is the therapeutic maneuvers. And again, we're looking at a scale of 1 to 5, 5 being comparable to a reusable device, and you can see most of the scores, the medians were either 5 or 4. So here's some representative images and videos, and I'll just sort of play one brief video here. This was a patient who had an anastomotic biliary stricture, from which had a metal stent, a covered metal stent that had migrated somewhat distally, and so that was removed. And then subsequently, we use a balloon catheter to drive a total of 3 stents across, and I found that the elevator is fairly robust in doing this. So you'll sort of see the first stent across, and then now you'll see the guide catheter for a second stent going up next to it, and a second stent being advanced, and then even a third stent. And again, I found that the ability of the stiffness of the device actually in some ways in the duodenum is helpful to help, sort of provide a firm scope to which you can drive across tight strictures. In fact, the very first case we did in humans, case number 1 in the trial, was actually a hilar cholangiocarcinoma, so which was successfully completed. So here's a third stent going across. One of the criticisms of our trial was that 2/3 of the patients had non-native papilla. So this was a randomized trial, 48 single-use, 50 reusable, all native papilla, most of which were in low complexity. Interestingly, these authors found they actually had a fewer number of attempts to cannulate with the single-use device compared to the reusable device. They did find it to be somewhat stiffer in terms of gastric passage. The image quality, they said, wasn't as good. There were some stability issues and some air-water button functionalities, which I think have largely been resolved, with the more recent versions. There's no difference in cannulation rates. Adverse events, need to cross over, or need for advanced cannulation techniques. And importantly, time to cannulate as well as procedure time were the same. They only had 2 crossovers out of the 48 single-use duodenoscopes, which again was very comparable to our data. This was a larger study that looked at with many of the same authors from that first study that I participated in, 14 experts and 5 less experts, although I would argue the less experts were all pretty expert here as well. So the 200 cases, again, 81 were ASG grade 3 or 4, and we actually, there were really no differences in the baseline complexity or any of the usual parameters. The SAE rate was about 6.5%. Interestingly, it didn't reach numerical significance, but you can see the crossover rate here was 18 out of 11% for the experts and only 2.5 for the less experts. So maybe the less experts felt like they had something to prove. I'm not sure, but in fact, they were trending better than the experts. And again, if you summarize this data from the 3 styles I just mentioned, as well as a European study, you got about almost 370 patients, success rates 91 to 97%, and comparable endoscopist satisfaction and adverse event rates. So when should you use a single-use device? This is probably one of the most common questions, and I would argue probably 4 buckets. 1, facilitate logistics and operations. Weekend and night cases, when you need a sterile scope in the OR, a backup fleet on a busy days, and perhaps as low volume or offsite centers where they don't have on-site reprocessing. You can also use it to protect the fleet in the case of a known patient with MDRO, as well as in critically ill patients who you feel couldn't withstand a scope-acquired infection. So transplant patients, ICU patients on pressor support, or even hospitalized patients in general. And then finally, in high-risk procedures for bacteremia, including cholangioscopy, biliary RFA, and stent placement, and perhaps even biliary brushings. So finally, some cost considerations. Obviously, healthcare quality is inversely proportional to cost, and there's several factors associated with owning a scope, whether you purchase or lease it, do you repair, what your repair costs, what your reprocessing costs, or what type of enhanced reprocessing are you doing, as well as the costs associated with potential litigation and intangible costs associated with a loss of reputation from an outbreak. So this is a crude study that sort of looked at, based on how many ERCPs you do a year, more to the right, and what your infection rate more as you go down, what would the price of the scope need to be to make it sort of economical? And so obviously if you do a lot of cases and you never have infections, the scope's got to be really cheap, like $109. And if you don't do very many, and you have a high infection rate, you might be willing to pay as much as $3,500 for these devices. And again, this is just a line graph sort of modeling the rates of infection. And they basically said a low volume center, scopes you need to be, could be up to $1,300. And at high volume centers, it needs to be $800 or less typically for this to work. Now, we did a study comparing the cost of single HLD to double HLD to ethylene oxide to a culture and quarantine approach, and basically found that given the delays in terms of cycle reprocessing and the need for more scopes, that the average high volume institution that does 1,200 cases a year might spend $300,000 to $400,000 more per year, primarily due to scopes, but partly due to reprocessing. And a medium volume 200 center ERCPs a year center might spend between $75,000 and $90,000 for that same amount. Okay, so this is a cost-effective analysis of single-use and reusable devices that we did. And again, so Markov model I did with an onion DOS, and essentially what we found is that given the fact that there is an outpatient payment now from Medicare through the transitional pass-through payment program, that when that occurs, you actually using a single-use device becomes dominant. It's the least costly and most effective option that is present. And the bottom line is that the price of doing ERCP is going to increase There's going to be new devices with scopes and adapters. There's new reprocessing technologies, new cleaning technologies, staff training and monitoring. And you're going to need tests for validation of reprocessing adequacy, all of which are going to add to costs. And of course, single-use devices are likely to add to that as well. There are some additional benefits of SUD. Obviously, as I mentioned, you may be able to, beyond infection control, you could preserve or expand the site of service to low-volume centers and small hospitals, ergonomic advantages, which we discussed, and I think a more rapid cycle of innovation. Rather than getting a new model like a car every 5 years for a scope, you might get one every year like an iPhone, because this project was pitched in 17. We did animal cases in 18. The first human case was 19. We're already on the third commercial iteration. And again, you may see specialty scopes for rare indications like thinner, longer, and rotatable scopes. So in closing, the potential for duodenoscope transmitted infections persists and continues to occur. Most outbreaks are due to inadequate recrossing in the region of the elevator, but there is some residual instrument channel contamination that can occur, and the potential for error in manual reprocessing in HLD is well documented, and single-use devices will eliminate the risk of device-transmitted patients to infections, and have shown promising initial results in clinical use in terms of efficacy. Reimbursement and continued price reductions due to economy of scale will be key in making utilization feasible, and patient preference will likely play an important role with respect to their future adoption. The benefits of single use may extend beyond infection prevention, and we do need further studies on the efficacy and cost effectiveness of these devices to compare them to low temperature sterilization or enhanced HLD to see which mechanism may be the ultimate solution. But I suspect a combination of these techniques will ultimately be used in clinical practice. Thank you again.